Hi guys, this is Chris. For our final project, we chose the article titled Engineered Human Myocardium with a Local Release of Angiogenic Proteins Improves Vascularization and Cardiac Function in Injured Rat Hearts by primary authors Fabiola Minarin and Rajiv J. Kant. According to the American Heart Association, approximately every 40 seconds an American will have a heart attack. This accumulates annually to about 720,000 new attacks in the United States alone, with about 14% of those leading to death. For the remaining 86% who do survive, these victims do not leave unscathed. Due to the period of the heart attack where blood supply is essentially stopped, myocardium cells permanently die and are replaced with scar tissue. This scar tissue, primarily made of fibrillar collagen, is not the best substitute for heart tissue, since it oftentimes fails and leads to further heart complications. To be more exact, nearly 1 in 5 people have a second heart attack within the first 5 years of their first because of the weak scar tissue. This tissue as well does not contract with the rest of the heart, which causes irregular and inefficient blood circulation. This can lead to several side effects such as shortness of breath, inability to exercise, fatigue, weakness, and an overall decrease in the quality of life. Solutions vary to help the healing process of scar tissue. Non-invasive options such as changing your lifestyle, including quitting smoking and exercising more, are commonly recommended. On the other hand, more invasive solutions such as ventricular restoration surgeries are also available. But due to the complexity of the problem and the sheer amount of patients who suffer from heart attacks, people are generally steered away from getting surgery. What is most promising, however, is the development of cardiac reprogramming technologies like what this paper explores. For biomaterial engineers, working on the heart is challenging since it's a highly specific and high-risk location if failure occurs. The scarred region also gets little to no oxygen or blood supply, making biocompatibility a struggle. However, this paper aimed to solve all these problems by creating an implant that provided full functional recovery of the heart through integration and vascularization of implanted engineered cardiac tissue.
Hello, my name is Ronak Bhagia, and I'll be walking you through the methods used in this study. There were 12 main parts. First, primary human umbilical vein endothelial cells, HUVECs, and normal human dermal fibroblasts, NHDFs, were cultured for the assays. Cardiomyocytes were differentiated and alginate microspheres were produced. This was followed by an in vitro biotin VEGF release study to quantify the release of growth factors from the alginate microspheres. These microspheres were loaded with the biotin VEGF and intubated, and then a Western blot analysis was performed. Infrared dye was used to detect biotin in the test samples, and a water control was included on every blot for standardization across tests. A two-dimensional network formation assay was then performed. Cells were treated with BFGF, VEGF, and combinations of the two in both low and high concentrations, as well as a control. Network formation was assessed for each treatment. Then, a 3D angiogenesis assay was performed. Collagen solution was mixed with the HUVECs for immobilization in the collagen scaffolds. Alginate microspheres containing different doses of growth factors were incorporated into the mixture. The test conditions included single, double, and triple combinations of BFGF, SHH, and VEGF loaded into the microspheres. Network area, number, and length were qualified from these images. Following this was the aortic ring assay. Wild-type rats were sacrificed and descending aortas dissected into thin rings. The rings mixed with the alginate microspheres, releasing various concentrations and combinations of VEGF, BFGF, and SHH. Cell migration and network formation were evaluated. Three-dimensional morphology and spatial organization of the newly formed networks were studied. The cardiac constructs were formed next. Cardiomyocyte morphology and function were assessed with acellular scaffolds produced as a control. Functional activity of the cardiomyocytes in the construct was performed by mechanical analysis. Active stress, upstroke velocity, time to 50% and 90% relaxation, stiffness, passive stress, and strain were all calculated, allowing for a determination of Young's modulus. With the in vitro aspects of the study done, the researchers moved into the in vivo procedures. Rats received a myocardial infarct. Those meeting study criteria were then randomly distributed into 4 groups. Group 1 was sham implants. Group 2, engineered constructs with only cardiomyocytes. Group 3 were engineered constructs with cardiomyocytes and unloaded alginate microspheres. And Group 4 was the engineered constructs with cardiomyocytes and alginate microspheres loaded with VEGF, BFGF, and SHH. The constructs were implanted. After 7 days or 1 month, the mice were sacrificed for study. First up was a histological assessment of the explanted tissues. For each heart, infarct area was quantified, cardiomyocyte morphology, tissue engraftment, macrophage infiltration, and vessel counts were examined. Then, a qualitative evaluation of vessel perfusion was done. Texas red tomatillo lectin was perfused into 4 hearts at sacrifice, each of which were sliced and imaged to create 3D reconstructed images. Following this was a quantification of angiogenesis by microflow perfusion, as well as a micro-CT imaging and 3D reconstruction of perfused vasculature. Micro-CT images were cross-referenced with the previously done histological images to identify remote, infarct, and implant regions. 3D reconstructions allowed for vessel analysis. This was used to quantify the number of vessels, vessel length, surface area, volume, diameter, tortuosity, and number of branch points in every region of interest. Finally, a statistical difference between the groups was evaluated with one-way and two-way ANOVA tests, followed by post hoc Turkey's or Bonferroni's test.
Hi everyone, my name is Caitlin and I'm going to walk you through the results of this study. First, researchers analyzed the release rate of the alginate microspheres and found that VEGF is released over 3 days. They saw that there was an initial burst where 50% of the material was released within 8 hours. Then there was a sustained release period for 72 hours, and finally no protein was detected after 7 days. Next, researchers found that growth factors VEGF and BFGF enhance endothelial network formation in 2D and 3D environments. The 2D vascular network formation assay was used to help determine concentration and combination of growth factors to use in the alginate microspheres, as seen in figures C and D. When treating endothelial cells, or ECs, with a combination of growth factors, angiogenesis, or the increase of network area and length, resulted. With this, researchers used the 3D network formation assay in figures E and F to isolate the effects of combinatorial growth factor stimulation on ECs. They found that angiogenesis was enhanced with a combination of growth factors released from low-density alginate microspheres, as seen in figure G. Researchers then looked at an aortic ring assay and found that heterocellular outgrowth from aortic rings is stimulated by growth factor releasing alginate microspheres. This assay validated the results obtained with the network assays in an ex vivo environment with heterogeneous native vascular cell populations. Cells from the aortic rings were able to invade the microspheres, and results were similar to the network formation assays as seen in figures B and C. Microspheres are denoted with yellow arrows. Researchers found that engineered cardiac tissue function was preserved with VEGF-releasing microspheres. Mechanics analysis demonstrated a significant increase in stiffness of high VEGF-loaded microsphere tissues when compared to the unloaded engineered tissues, as shown in figure B. The peak active stress was also significantly higher, as seen in figure C. Finally, researchers determined that release of growth factors from cardiac constructs promotes neovascularization in the implants and improved heart function in a rat model. Cardiac function was significantly higher in the groups implanted with the experimental tissues. Automated 3D analysis of networks traced in the implant volumes showed an increased number of perfused vessels and presence of larger vessels with higher number of branches, as shown in figures B, C, and D. Typically, larger vessels give rise to more branches, and the stronger linear regression between vessel diameter and number of branch nodes for the loaded implants suggests that more complex network structures formed, better resembling the architecture of the natural cardiac system.
Hey guys, my name is Ian. Biomaterials have had wide application to the field of cardiac tissue engineering due to their ability to store and release biological compounds such as growth factors and proteins. In this study, a novel method of promoting revascularization in rats who had suffered myocardial infarction was demonstrated. In doing so, the researchers tackled many of the problems relevant to the field at large. For instance, a controlled release strategy is necessary when delivering angiogenic growth factors due to their short biological half-life in situ. However, the prolonged and continuous release of growth factors have been shown to induce the growth of vascular tumors. In order to account for this, the biomaterial presented by the researchers releases VEGF and BFGF in a temporally restricted manner over the course of 3 days. The ability to fine-tune the period of release of a drug from a biomaterial is tantamount toward developing new advancements in cardiac regeneration. In addition to a controlled release strategy, the researchers also made use of alginate microspheres as local protein deposits. Alginate, a compound derived from the cell walls of brown algae, has been used previously as a biomaterial for cardiac regeneration. However, in this study, alginate was specifically used to promote healing following myocardial infarction. The researchers demonstrated that the delivery of immunomodulatory cues via alginate microspheres efficiently triggered phenotypic changes in the cell population, simulating tissue repair in the process. Furthermore, the engineered myocardium was able to incorporate the microspheres without major deficits in mechanical properties. These findings paved the way towards the future use of alginate in clinical applications and biomaterials research. The study also suggests possible avenues by which research on this topic may proceed. For instance, the specific pathways by which VEGF influences engineered cells remain unclear. This relationship is important to understand because the stiffness over time of the engineered tissue depends on the concentration of VEGF. A major limitation of the study, as well as similar studies, is the persistence of fibrotic scar tissue in the affected region of the heart. The presence of scar tissue may adversely affect the revascularization and regrowth process. In order to bypass this obstacle, research into methods of scar reduction or research into growth factors which have a higher compatibility with scar tissue is key. In total, the research highlighted the ability of smart biomaterials to stimulate significant regrowth of heart tissue following myocardial infarction, providing a foundation for the development of combined regeneration and revascularization techniques in the future. Overall, the study is relevant to the treatment of heart attacks. Not only does the development of engineered cardiac tissue help doctors to treat heart attacks in a more advanced and effective manner, but the research also increases the collective understanding of how biomaterials can be used to solve problems in cardiology.
The methodology of the study seems solid. The researchers conducted a thorough in vitro exploration of the alginate microspheres before moving on to the in vivo mice models. Positive and negative controls for both the microspheres and the engineered constructs were used, and the study included controls to normalize results across different tests and between different researchers of the team. This ensured the statistical analysis in the results section was as sound as possible, despite the numerous variables involved.
The only current treatments to help restore the heart that I have heard of involve surgery, which is not ideal for a patient. If this study proves successful in later stages of research, it would be a preferable method.
However promising the study is, there are a lot of steps to be taken until these technologies are used in humans. In order to further this product down this pipeline, human clinical trials should be approached next, and further refinement of the implant should be conducted. This paper showed the cusp of what is to come in treating heart disease, and it's exciting to see where it will go.